Hey guys, this is a little bit more of a little bit of detail on some of the different BLAST outcomes, so you guys can kind of get a sense of how that works and what they do. So the first thing I want to do really quick is just spend a minute or two on what these things are. So nucleotide BLAST or BLAST-N is obviously looking for nucleotide similarities from your query to all the subjects that are in the database. This one, BLAST-P, is looking at protein sequences, so basically amino acid sequences comparing against other amino acids. BLAST-X is when you have some DNA and you want to see what sort of protein sequences it might align to. And then there's also a couple of other ones that are on here. There's a T-BLAST-X, which is a little deeper, so if you end up finding that one, that's just a way to find very distant relationships between nucleotide sequences. So let's just do a regular one here. And let's go to hit our regular nucleotide BLAST, and I just have a little segment of DNA here. So let's just say that this is from some sort of paper that it's a primer sequence, let's say, for PCR, and you want to see what this thing might help to amplify in a conventional PCR. So I go ahead and hit my BLAST, and let's see what we get. Okay, so here is the results that we see. And when we see this, let me make the screen a little bit bigger for you. There we go. When we see this, you'll notice that there we go, that it looks like something called Ehrlichia chaffiensis. That's a bacteria that's found in some ticks that can cause ehrlichiosis. So what's all this mean? Well, basically, our query came back with a lot of matches to things in the database called the subjects. And you can see, and the good news is that almost all of these, to this point, are Ehrlichia chaffiensis. And then we start having it connect with things that aren't Ehrlichia chaffiensis. And so how are they organized? Well, you can see here that, first of all, it's by the best fit is first. And so you can see right now we have what's called max score in the corner. So first of all, we have the scientific name of the organism, and then we have something called max score right here. And basically what that is, that's a number of the matched bases in the longest highest similarity pair of this particular match that we have. And that sounds a little tricky, and we're going to get to a little more detail that in another section. But really what it means is the higher the better. And the same with the total score, the higher the better. Then you get to query cover. Now this is important. So query cover basically means that what percent of all of the number of nucleotides match the query and the subject. So for example, if your query that you inputted had, let's just say, 18 nucleotides, if all 18 nucleotides are matched to the subject, then it's 100%. But what if you only put, what if your query had 27 nucleotides and somebody ended, their submission of GenBank where only about 15 of them overlapped, something like that? They wouldn't all overlap, and I'll show you kind of what that looks like. So that's what percent of the query cover is also queried by the subject. And then the next one over there is called the E-value. And that's basically a statistical tool. And really what it says, it's the number of matches you would expect to find by sheer chance. Almost like, in essence, like a hypothesis test of some kind. And you can see that it's very low and you want it to be low. So in this case, smaller is better. And you can see it's 0.001% chance that these would align up by sheer chance. And so that's really, really good. And then percent identity would be, out of all the nucleotides in the query and the subject, how many of them are letter for letter? Like if your query was AACAC, then the subject was AACAC, and that's 100% identity. Then, of course, we have the accession information, which is the codes to be able to find. First of all, this is the accession length, so this would be the generic. Sorry about that. So basically, the accession length is the number of the nucleotides. Or again, if it's BLAST-P, it'd be the number of amino acids. But in this case, it's the number of nucleotides in the result subjects that are identified by the actual session number. So this subject has, it's part of a really long piece, and it makes sense because this first one is the entire genome of Ehrlichia chaffiensis, and therefore that sample is 1,170,935 base pairs long. And you can see these are all genomes. That's why they're the length. And we get down here, we get into smaller pieces that people did. So that kind of gives you an alignment. And the good thing is if you were trying to do this for something like PCR, this would be a good primer to use, in part because you can see it delivers 100% query and 100% identity with very low e-values for every single thing that's Ehrlichia Jaffiensis. So let me just do one. Let's just compare two things. This thing, and let's go down to something that's not 100% covered, and this thing, and we're going to do an alignment. So what we see here is the first example is there's our query that we put in. That's 26 base pairs, and this is the subject it found. And you can see that all 26 match them 100%. Everything's lined up. There's no gaps, meaning that there's no indels or any sort of insertion or deletion or blank spaces. And so this is really good. So now we see the other one I chose. And this one has a lower score, first of all, right? And and what we end up getting is a different combination of potential identity. We see that there's a difference here, an A and a G, and so that's going to lower the identity a bit, and so that would make it a lot less likely to be obviously number one the same thing, but if you were using PCR it wouldn't be as efficient. So that's kind of the idea of what what those numbers mean when you actually get the results back of all of the other things. And another neat little tool to look at some stuff is something called taxonomy. And so this is, you can also, by the way, I coded this before, but you can also just kind of, from the one sequence that I selected, you can just click on taxonomy and it'll tell you all about what that is. And so if I'm sitting here, doing all this stuff and I'm like, what is this? I've never heard of Asapenser ruthiensis, which I've never heard of it. You can just hit taxonomy and it'll tell you exactly what it is. And so if I were to then do this and be like, what the heck is Ehrlichia Jaffiensis if I've never heard of it? It's a bacteria. And you can actually click on the description of it and it'll tell you all about it if you wanted to read all this stuff, and that's great. So that's just a little bit of an introduction to the results that you would see when you do a BLAST-N.